Hi, the purpose of this presentation is to give you some information about cell membranes before you get to class. By the end of this PowerPoint, you want to be able to draw and label the structure of the cell membrane, identify the major functions of the cell membrane, and discuss the proteins that exist within the cell membrane. This is a picture of a cell membrane, and it probably looks a little bit different than you remember from freshman year. We kept it pretty simple, but it is a complex set of molecules, and each of the molecules have specialized functions. Let me point a few of them out to you. If you remember that the phospholipid bilayer is basically how we describe the structure of the cell membrane, what I circled for you is a phospholipid. So on the right of your note sheet, I say draw a phospholipid. Draw one of these guys. Okay. We'll talk about what that is in just a minute. These purple things here are proteins. Notice some of the proteins go the entire length of the membrane, and some of them only stick out the top, or they would stick out the bottom. Okay. There are yellow molecules here, that's cholesterol. Cholesterol helps with the flexibility of the membrane. We have glycolipids. If you'll notice that this is here is a carbohydrate, and then this is a fatty acid. So together it's a glycolipid. Over here you have a carbohydrate and a protein. So we call those glycoproteins. So there are a lot of different molecules that hang out next to each other, and that's what makes our membrane. This is a really neat image of a cell membrane with an electron microscope. So what we see here is this would be the one side of the membrane, this would be the other side of the membrane. And so inside here would be your fatty acid tails. So your phospholipid, so your phosphate head would be here, and your tails would be pointing towards the center. So that's what that would look like under a microscope. So like I said before, the cell membrane is a phospholipid, and it's a bilayer because there are two layers. What it really is, it's a triglyceride, and it is missing a fatty acid, and in place of that third fatty acid we put a phosphate group. So this phosphate group right here is polar, and the fatty acid tails, of course, are nonpolar. And so that helps with its function, because when we make a cell membrane, these fatty acids point to the center and the phosphate heads point to the outside. And so that's going to help repel most substances from going across the membrane that are polar. And then because those molecules are so close together, most molecules aren't going to be able to fit through the membrane. Alright, so here is the molecular structure of a phospholipid. This is a P, really. So here's your phospholipid head, or sorry, phosphate head. And notice it's polar. Here is your glycerol. And here are your two fatty acids. Before, when we drew a triglyceride, there was a third fatty acid here. We don't have that anymore. And what happens is we replace those extra electrons with a phosphate head. So it's two layers. And I said this before, and I'm going to tell you again, that those molecules aren't bonded together. They kind of just hang out. What I like to picture is if you were at a pool party and there are a lot of people with noodles and rafts and tubes hanging out in the wave pool. That's what it's like, is that you're kind of all stuck together because you're in a confined space, but you're not bonded together. So where it tells you to draw the cell membrane, go ahead and draw this simple structure, and you can go ahead and pause this presentation if you want. Make sure that you label these as polar and these as nonpolar. It's a bilayer because you have one layer here and one layer here, so there's two layers for you. So these are your fatty acid tails, and these are your phosphate heads. This is a neat image because what they've done is they've frozen cell membrane, and then they took the top half and the bottom half. So both of those, since it's a bilayer, they've basically split open the bilayer, and then they look under a microscope. And you can see that in the inside, there's not as many dots, but in the inside of the cytoplasmic layer, there's a lot of dots there. And what do you think those dots are? If you look over at the cartoon here, can you tell what the dots are? Yeah, they're proteins. So when you look at the layer that's closer to the actual cytoplasm of a cell, it's going to have more proteins than the actual outside. And that makes sense because proteins help the cell communicate and help it do different functions. So it makes sense to have them close to the inside of the cell. Because it's not easy to see the cell membrane, we come up with a model. How does the cell membrane act within all of the different molecules? And so the model that they come up with is called the fluid mosaic model. Okay? If you think of the word fluid, it means free floating, that everything kind of is moving. And if you think about a mosaic, what do you think about? A lot of times I think of one of those stepping stones where you take a lot of different broken plates and you put them down, so you make some type of picture out of different pieces, or maybe like stained glass, some type of artwork where it's a mosaic. It's made out of different materials that makes a picture. So they say the cell membrane is a mosaic because there's lots of different molecules that make it up. So we've talked about those, the phospholipids, cholesterol, proteins, your glycoproteins, and glycolipids. And then the reason that we say it's fluid is because those molecules aren't bonded together. They just kind of float around each other. So you can kind of think of it like tiles that are in grout. But eventually, what does your grout do? Your grout hardens, right? And so then those tiles aren't floating. So again, going back to my pool party analogy, I like the idea where if you have basically your phospholipid bilayer, which would be your water, and then your pool noodle, your tube, your raft, those would be the different types of proteins, cholesterol, all that kind of stuff that are floating within the water, or your proteins that are floating within the grout. All right, so we have this picture here on your notes. So why don't you go ahead and label this now. So this is a protein. This is cholesterol. This is your glycolipid. This is a glycoprotein. And of course, this is a phospholipid. So again, if you want to pause it so you can label those with the correct spelling, go ahead. Otherwise, we'll go on. So the cell membrane has a lot of different functions. The individual molecules also have functions. So the phospholipids we talked about, again, structure, function. Their structure, the inside is nonpolar, and then the outside is polar. So that's going to create a barrier for water, other polar molecules. The cholesterol creates flexibility in the membrane, so if the membrane is touched, it's not going to pop. There's some spring action there, and it basically just has flexibility. We'll talk about proteins in a minute. And then glycoproteins and glycolipids, they label the cell. They're like name tags for the cells. And so when one cell bumps into another, they see those glycoproteins or glycolipids, and they know what type of cell that is. That's also could be the way the immune system could help identify invaders where they wouldn't recognize a glycoprotein or glycolipid on a foreign cell. So I want you to answer the next couple questions. So can membranes, lipids rotate? So if you have the phospholipid, can it rotate around, like spin around in a circle? Can it flip from one side of the membrane to the other? And can it move from one place in the membrane to the other? Go ahead and write down your answers. All right, so check your answers. Were you right? Were you wrong? Do you understand why the answers are what they are? All right, so the cell membrane as a whole controls what goes in and out of the cell. It allows communication between cells. It keeps the cell's contents isolated from the external environment. All right, so remembering, the outside is polar or hydrophilic, the inside is hydrophobic or nonpolar. So lipids can get through if they're small enough, but since sugar will probably have polar places, it's not going to be able to get through this membrane because this section here is nonpolar. Water, a lot of times, can sometimes sneak through, but for the most part it can't get through the membrane. Salts have charges the same way. Waste down here, like urea, won't be able to get through because it's too big. So the cell membrane is really regulating what goes in and out. It's, it's like somebody standing at the door telling you if you can come in or not. Okay. So these different proteins within the membrane, they have different functions too. So there are a couple different types of membrane proteins you want to make sure you know. So there's peripheral proteins, and those are the ones that are on the surface of the membranes. So like it's circled right here, and it doesn't go all the way through. This one, an integral protein, would be this one here that goes all the way through the membrane from one side to another. So peripheral proteins are going to be like name tags. These integral proteins a lot of times are channels, kind of like tubes that allow things to go from one side to the other. Or if the cell is trying to communicate with another cell, it would want to go all the way through, so that signal could be sent from the cytoplasm of a cell across the membrane to touch the membrane of another cell. These integral proteins are also called transmembrane proteins. Remember, trans means across. Alright, and so our proteins have a couple different jobs. So I like to put them into these 5 categories: a transporter, enzymes, receptors, recognition proteins, and adhesion proteins. And if you just look at the word, you should hopefully determine, for the most part, what the protein is. A transporter protein can transport substances across the membrane. That's seen here. We've talked about enzymes. They regulate chemical reactions. So they're either a lot of times gonna either synthesize or decompose a molecule. There are receptors. So those receptors, a lot of times it'll be a molecule that when it attaches to a protein, it sends some type of signal into the cell. We talked about cells as name tags, okay, or recognition. And then it can also help us attach to each other. So if you think about your skin cells, how are they staying connected to each other? Well, we have proteins that connect our cells together. So hopefully, if you just look at the word, you should think, okay, transporter is going to transport molecules across the membrane. Enzymes, chemical reactions on either the outside or the inside of the membrane. Receptor. Okay. So receptor, they receive a molecule to tell the cell to do something. Recognition. Oh, I recognize you. It's your name tag. Adhesive and adhesion, they stick together. All right, and for the rest of these are just some examples that we can go through to reinforce the different types of proteins. Transport proteins, again, they're going to regulate different molecules across the membrane. There's channel and carrier. Channel proteins are usually diffusion, carrier active transport. There's a special molecule protein called an aquaporin. What do you think it transports? Yep, it transports water. So each protein has a particular shape, and remember, its shape has to be dead on for it to have a particular function. All right, so the last thing we'll talk about in the last 30 seconds here are these proteins. Remember, in order to be here, they would have to have the correct charges. So the charges of the amino acids here would have to be somewhat nonpolar in order for them to be able to be here. The charges of this and these R groups would have to be polar because they're sticking on the outside towards the water. If you have any questions, please make sure that you ask your teacher, and that's all for today.